to thank the organizers for their persistence in organizing and reorganizing this event, I think 3 times at least. So it's great that it's coming off virtually. So our group is focused on trying to understand physical principles involved in genome organization. And I'm going to tell you a little bit about how genomes are, we think, compacted more than condensed during mitosis. And what we think some of the mechanisms are underlying that, including loop extrusion by SMC complexes. And also talk a little bit about a topic closely related to the last talk, concerning the organization of the interphase nucleus, where it looks like HP1 alpha plays a key role as a kind of mechanical as well as genetic control element. So I'll start talking a little bit about metaphase chromosomes. There's a kind of representation of a cell cycle in a eukaryote cell where we completely geometrically reorganize chromosomes as we go from interphase to metaphase and back through DNA replication. And what's really remarkable about metaphase chromosomes, well, 2 things. They're separated from each other, and even the chromatids inside them are separated from each other, despite being close to one another. And they're not droplets. They're long noodles. And this is already very surprising because we often use the term chromosome condensation to talk about metaphase chromosomes, and I'm going to try to convince you that that is a very bad term to use. All right, so what we know historically are many things about metaphase chromosomes, but perhaps one of the most important results were results of studies of Ulrich Lemley that really established that chromosomes at metaphase are organized into a tandem array of loops of chromatin, and that we see them as solid objects, but they're actually a bunch of loops that are packed together along the axis of the chromosome with a condensin-rich axis along the chromosome. This was already established in the early 2000s by Lemley's group. And you can kind of see this brush organization. And classically, it had long been thought that the axis along the metaphase chromosome was a kind of contiguous proteinaceous structure. But you can even see here that there are really clusters of these black dots, which are immunogold targeted to SMC2, targeted to condense. So one of the ways we've approached looking at organization of chromosomes has been to actually play with them and to take genomes. In this case, this series of images are tissue culture cell growing at metaphase. We open the cell, take the genome out, isolate a chromosome, and take it away for physical study. And this can be done in combination with genetic or biochemical perturbations and can provide structural information through the age-old method of material science of looking at what the mechanical properties are of a component of the cell. So the native metaphase chromosomes, we've studied them from many different types of eukaryote species, and they are nice elastic objects that show a nice reversible elasticity. And here, this assay uses one pipette attached to one end of a chromosome to pull the chromosome and stretch it while watching the other pipette bend, and we can calibrate that bending stiffness and measure the mechanical response of the chromosome. And mitotic chromosomes are nice reversible springs as long as you don't stretch them too fast or too far. And so historically, we started with newt chromosomes. We now exclusively study mammalian chromosomes, human and mouse chromosomes. And of course, newt chromosomes, to double them in length, take a big force of about a nanonewton. Human chromosomes, to increase their lengths, take much lower forces in the tens of piconewtons range. But they both have about the same Young's modulus, a few hundred Pascal, okay? And I won't talk about viscosity, but the internal viscosity of these structures is tremendously large, as we would expect from a polymer material, but where the polymers are able to move around quite freely. So a remarkable and old experiment from our group done by Mike Poyer at the beginning of these studies involved taking a newt chromosome and then spraying it with a condensing agent. in this case, cobalt hexamine trichloride, which strongly condenses DNA and chromatin, classically used condensing agent. And what happens in that case is that the volume of a metaphase chromosome goes down by about 2/3. This suggests that at least 2/3 of the volume that we see in a metaphase chromosome is actually solvated volume, and that only 1/3 of the volume of a metaphase chromosome is actually sort of solid chromatin. Again, suggesting against the idea of a strongly condensed structure. By the way, the stiffness of this condensed chromosome is gigantic. So if we take a metaphase chromosome and expose it to restriction enzymes, which cut periodically at some statistical frequency between nucleosomes, in this case, this is ALU1, which cuts every 256 bases statistically in random DNA, but in chromatin, where 90% of the DNA is covered, is protected by nucleosomes, it's something more like a couple of kilobases. And the nut chromosome is cut through, and you see it changes from being a solid structure to a liquid structure, and actually that liquid droplet will slowly evaporate. For a human chromosome, that kind of digestion experiment causes the whole chromosome to kind of disappear. So what our picture is of the interior of the metaphase chromosome is some kind of cross-linked array of regions with some kind of linkers, presumably proteins and presumably condensin, which play the role of holding the chromatin together into this noodle-shaped structure. And then you could ask, are the condensin SMCs the major cross-linkers of the chromosome? So if we take a metaphase chromosome and spray it with antibodies for SMC2 and then use secondaries, before stretching it out, it looks like what you would see in any cell biology experiment, a kind of a track of SMC2 along the chromatids of each chromosome. But if you stretch the chromosome out, you see that the SMC2 rich region has many gaps along its length. And this is consistent studies in other groups, which really ultimately see a disconnected SMC2 structure in a condensin structure in the interior of the metaphase chromosome. Well, and we've done similar experiments thanks to the Takagi lab, which has made cell lines that carry GFP condensin fusion proteins in them. We can actually do the same experiment with intrinsic labeling, and again, you can see this variability in the amount of condensin along the length of a chromosome, independent of the of things that might confound us antibody spraying experiment. So we think the SMC2 rich region in the chromosome is actually disconnected. And how can that be? How can you get a noodle-shaped structure with disconnected SMCs that aren't stuck together into a kind of a scaffold? Well, okay, in the same kinds of experiments we've done, we've indeed seen that if we siRNA out the SMC2, we see the stiffness of the chromosome go down, as we'd expect if it played the role of a cross-linker. And we see that if we stall the cells in mitosis using colchicine or nicotazole, the amount of SMC2 goes way up on the chromosome, and you see it's much stiffer. So SMC2 and condensin does appear to play the role of a kind of a crosslink. If you plot the fluorescence in those experiments versus modulus for a bunch of individual experiments, you get a nice continuous relation showing that the more SMC2 there is, the more stiffness you get. And incidentally, in the colchicine-treated cells, when you stretch the chromosomes out, you still see this punctate condensin distribution along the length of the chromosome, but a lot more condensin near the centromere. So how can this work? Well, what we've hypothesized for some years, and this is a result of a computational study done in collaboration with Leonid Murney, done by Anton Golubadorko, what we suppose is that an initially interphase chromosome, which is not folded into a metaphase chromosome, if it's acted on only by loop extruding enzymes that are able to bind, pump for a while, pump loops out for a while, and then dissociate, that turns out to be sufficient because of crowding of the chromatin and crowding of the SMC complexes along the length of the chromatids to reorganize a chromatid into a superpolymer, which is much stiffer than the underlying chromatin polymer, and has a well-defined thickness. And the only way this can work is if the chromatin doesn't stick to itself, so that we gain the osmotic pressure from the loop pushed together, which define the chromosome axis. Okay, so this type of linear chromatin structure we see in higher eukaryotes is intimately dependent on not too much sticking. You might be able to get away with a little bit of sticking of nucleosomes to each other. They can't stick to each other. And of course, if they did, all of the chromatins would stick together and all the chromosomes would stick together. So we should not use the word condensation. There's no condensation in this study. There are actually only repulsive interactions and the motor activity of the loop extruding enzymes. In fact, this is without traditional cross-linking or condensation, compaction. You can also see that there are gaps between the emergent regions of SMC2 rich chromatin, similar to what we've observed in experiments. Okay, so just to emphasize the role of the non-condensation effects and the excluded volume effects, if you stick two chromatids together and let this system loose on it, organizing loops by loop extrusion, what happens is, of course, the two polymers, as extrusion occurs along each polymer, push each other apart. In these experiments, there's effectively topoisomerase. In this computer experiment, there's effectively topoisomerase acting, which is allowing the blue and red polymers to pass through each other without any preference built into the topoisomerase. Driven by osmotic pressure, the two chromatids push each other apart, and you end up with something that looks like a well-resolved metaphase pair of chromatids. They're actually trying to get away from each other and making this kind of nice X shape that you see, particularly in meiosis II, in meiosis metaphase. All right, good. So it turns out that if you make a simple model of a polymer brush based on this kind of thinking with no attractive interactions, you can actually explain the length of metaphase chromosomes as a function of chromosome size, essentially genome size divided by the number of chromosomes in the genome, across a wide array of species. In fact, Eric Kramer, a colleague of ours at Simon Rock College near New York City, has recently done a big study of fitting the power law that's expected from a polymer brush model for metaphase chromosome to a wide variety of data across many species. You can see this is C number of species divided by number of chromosomes. And this power law works pretty well. I won't go into all of the details of predictions of the model. This seems to work remarkably well. So polymer brush picture of the metaphase chromosome is starting to look like a pretty good model for predicting the sort of spatial organization. So how do SMC complexes work? Well, I wanted to talk briefly about our ideas developed by collaboration with Paulo de los Rios and Alessandro Barducci, but particularly with Stefan Gruber, who's an expert on bacterial SMC complexes and is also very interested in the relationships between bacterial and eukaryote condensins. And so what we know about condensins and other SMC complexes is that they're ATPases and they go through an ATP binding and burning and release cycle. And the conformation of the SMC loop, these are the long SMC proteins in this little sketch here, and this is supposed to be the glycan and other accessory proteins associated with the SMC complex. When ATP binds, the two Walker domains can be bridged, dividing this protein loop into two compartments, and then hydrolysis opens those compartments. Coupling DNA to this ATP cycle of the enzyme, we know that from many studies, including those of Christian Herring and Kim Naismith, the DNA is often threaded through the SMC complexes. And if we imagine that the DNA is held down here in this lower domain, then when and this upper loop is essentially closed, the two protocols are closed. When binding occurs of ATP, it has been observed that this compartment can open in many SMC complexes, allowing a DNA loop to come in here and be inside this protein loop. Then hydrolysis, burning of ATP, releases the DNA, which is bound at this Walker bridge. And now you're left with this piece of DNA that can pop through. Then when this thing closes back down, you can get back to the original state, but translated along the DNA. So this is a model for translocation of SMC complexes along DNA. And it predicts a very low stall force in that sort of half a piconewton range, which is similar to that that's seen for mechanical experiments, single molecule experiments on condensing molecules. So you might want to know, like, can this kind of a mechanism actually work? So in collaboration with Enrico Carlon's group, and particularly Stefanos Nomidis, we built up some MD simulations of this model. And here's the translocation occurring, and you can see the DNA being bound in a loop after ATP binding, which opens the coiled coils, and then the burning of the ATP releases this chunk of DNA from the bottom, and you end up with the SMC complex that's translocated along the DNA. And you'll see there's ATP hydrolysis, the thing closes down and pushes the DNA entropically back into the bottom of the SMC complex. And so you started here, and you've come over here. And so we have a lot of ideas about how this basic model can occur, but it depends on a conformational change, which is polarized along the SMC complex. Of course, the DNA is unpolarized, so you have to have some way to remember what direction you're going along the DNA. And the SMC complexes have that built into their structure. So this bends, pushes the DNA into the upper compartment when it opens, and then ATP burning allows that DNA. So will this work for loop extrusion? And it turns out it will. And so here's a movie, again, for molecular dynamics simulations, done as realistically as we can on this kind of time scale, whereby you see successive enlarging of a loop of DNA, which is now essentially the SMC complex translocating along the DNA, which is looped back and attached to the outside, in this case, of the SMC complex. This is a feature of yeast condensin, which is known. There's a so-called safety belt domain that is able to trap DNA on the outside of the enzyme, which has been documented by Case Decker and Chris. What's remarkable about this is you can see that the topological memory of having the DNA threaded through the SMC complex allows this thing to translocate even when you have a complicated potential for sticking of the DNA to many different places on the outside of the protein loop. The fact that the DNA is on the inside of the protein loop guarantees that it can translocate even in this complicated geometry. You can see the loop gradually growing. And this will work along chromatin. I'm not going to dwell on this, but the hole in our model SMC complexes and in real SMC complexes is large enough to accommodate a ball the size of a nucleosome going through the SMC complex. That doesn't mean that nucleosomes might not be destabilized during SMC translocation along the DNA, but it's not really necessary. All right, so what I wanted to finish with was to talk a little bit about the mechanics of the interphase nucleus, and this is a pre-replicative nucleus work. We've heard a lot of discussion about organization of it this morning, and Andrew Stevens in my lab, who's now moved to UMass Amherst, the future tense here is current tense is present tense is maybe not, it should be changed to past tense. He's now setting up his lab at UMass Amherst, and he developed a method in our lab for carrying out the same kind of experiments I showed you for metaphase chromosomes on isolated eukaryote nuclei. And so the idea is to attach a whole nucleus to two pipettes and look at the mechanical response. And what Drew found was quite interesting. So he set up experiments where basically you pull on one pipette and watch the nucleus stretch, and it plays a role of the spring, and you can measure the tension in that spring, transduced to a second spring, which is the second pipette. And actually, we had a wonderful collaboration with Bob Goldman's lab here at Northwestern. They're a huge expert, of course, on the nuclear, and in particular, nuclear lab. All right, so what do we see? So what Drew saw in those experiments was that there are really two regimes of stress-strain response. So for short strains, up to a couple of microns for a human nucleus, that's sort of about 10 to 20%. You have a kind of a initial linear response. There's very sluggish relaxation, and you can go out again along the same linear response. We can do this in the cell. We can also do this for isolated nuclei, and we get similar kinds of force responses. You can grab the whole nucleus actually inside the cell for that. Now, only for nuclei taken outside the cell we can do larger deformations. And what we see is evidence for a secondary regime where the initial force goes up with one spring constant to about 10 or 20% stretching. And then beyond that, we see a stiffer secondary response. And of course, the nucleus is a lot more complicated than a metaphase chromosome, the kind of a multi-element mechanical system. So this second spring constant is seen to be larger in a series of different types of mammalian cells. All right, so what we can do is we can take a nucleus and treat it with restriction enzyme, and that will reduce that initial spring constant. But perhaps the most impressive experiment of this type is to take a nucleus, which we initially measure the spring constant of by stretching it, and then we treat it with micrococcal nuclease. And in that experiment, the nucleus starts out being kind of a ball that you can see. And at the end of the digestion, there's sort of nothing left that you can see because all the chromatin has been digested and into tiny bits of chromatin that have left the nucleus. You can see there's very little phase contrast. And we can still stretch out that structure. It has no spring constant at all, up to about 2 microns of stretching, but then it has the same spring constant as it had in the native state. So this indicates to us that the chromatin plays a key role in providing the mechanical resistance of the nucleus against any mechanical stress put on it in the cell. But then there's something else, and it's the nuclear envelope. That's all that's left here, which is providing this secondary stretching. And we've done separate experiments I won't go through on lamin mutants, which show that that secondary stretching regime is changed in, for example, lamin A mutants and not. So, and in fact, Ed Bannigan and our group did very nice simulations to show that the wild type behavior of this type of system, the simulation involving a polymer in the interior and a polymer shell in the exterior. The polymer in the interior is cross-linked to start with, and cross-linked to the exterior polymer layer, which is also a 2D cross-link layer. And we put in spring constants, which we think are representative of what happens in the experiment. You can nicely see this 2 spring constant behavior for an interface nucleus. If we take away the interior polymer, we have, again, no stretch, no stiffness at all, up to a threshold of around 20%. And then we see the external layer stiffness. And so this type of simple model has provided a nice framework to think about these types of experiments. What I want to point out is that this 2 spring constant behavior, the data aren't shown here, but the 2 spring constant behavior depends on the attachment of the interior polymer to the surface. If you don't attach them together, you just see the shell and not the polymer in the interior. All right, so once we know about this kind of thing, okay, so we have this 2-element system with surface elasticity and bulk elasticity, the bulk elasticity playing a role for initial deformation, the surface elasticity playing a key role for large deformations beyond 20%. Well, if we start to destabilize the chromatin, and in particular heterochromatin in the interior of the nucleus, can we destabilize the nucleus as a whole? And these pictures sort of show the story. There's some wild-type nuclei. These are nuclei where there aren't lamin defects, but we've introduced a reduction in the amount of heterochromatin by reducing the amount of histone heterochromatin mark methylation. And what you see is you can see lots of, this is a cartoon, but you see lots of blebs in the lower diagram. Okay, so the idea is we start with wild-type nucleus over here. We can push it towards being euchromatin by either cranking down histone methylation or cranking up histone acetylation, for example, using HDAC inhibitors. Or we can push the nucleus towards being highly heterochromatinized by using epigenetic drugs like methylase inhibitors. And so what we see is indeed treatments of wild-type cells is just representative curves, but when you do the statistics on a bunch of measurements of wild-type cells or cells treated with HDAC inhibitors like VPA and TSA, we see a reduction in the initial chromatin elasticity. And if we treat cells with epigenetic drugs such as methylstat, which is a Jumanji inhibitor, we see an increase in the stiffness. nuclei. Notice that there's quite a lot of dispersion in those elasticity measurements, so you have to do a number of experiments to get a population average for them. So, and there's the same kind of experiments done with yet another type of epigenetic drug, which increases which increases the amount of euchromatin. So, what's really remarkable is how much decreasing the amount of heterochromatin, increasing the amount of euchromatin, leads to increases in blebbed nuclei which have herniated region in them, which is a potential site for nuclear rupture. So here are the wild-type cells, and here are BPA-treated TSA and another drug-treated MEFs and HT1080 cells. You see it's a big increase in the bleb scoring. These blebs tend to be along the major axis, suggesting that they occur preferentially here in the regions of highest curvature. So they seem to be mechanically correlated, or at least structurally correlated, with regions of highest curvature. And this movie was just showing a actual bleb event where we can monitor disgorging of the nuclear contents using using nuclear localization. Okay, so a key question is, okay, so the changing histone marks can change mechanics of the nucleus and stability of the nucleus. So what is the mechanism of response to changes in histone marks? And so we were fortunate to work with a bunch of people, including Gradine Lab out in California, the Hutch, who developed HP1 alpha degron system, which allows us to rapidly take away HP1 alpha, which is, of course, one of the readers of H3K9 multimethylation marks involved in constitutive heterochromatin, as well as being a target of a lot of interest because of its phase separation properties observed in vitro. And so there's the HP1 alpha, which is fused to a fluorescent reporter in these cell lines, and the cells grow happily until you treat them with auxin, and then the nuclei become funny shaped. Here's the scoring with auxin treatment, an increase in amount of irregular shaped nuclei, which can be rescued a little bit using over-methylation. Kind of interesting that you can take the HP1 alpha away, but you can rescue the irregularity of the nucleus and make it more regular by taking by adding a lot more methylation to the histone. And the nuclear spring constant shows concomitant consistent trends in that when we treat with auxin, taking the HP1 alpha away, we see a big hit in the nuclear spring constant, nanonewtons per micron there. And then if we treat with methylstat, we can rescue some of that spring constant, but we can't drive the spring constant up to the levels that we would see by just over-methylation. So this is recouping the early experiment. So this is a great collaboration. The lead author, Amy Storm from Cliff Brangman's lab, did a ton of cell biology. Also, the Gradine lab did all of this nice genetics for us. Andrew Stevens is the lead author. It's his sort of crowning glory from his time in my lab. So this establishes that HP1 alpha is really responsible for reading those methylation marks and helping to control nuclear shape and nuclear stiffness. I don't have time to tell you about a regulatory pathway that Andrew found, whereby actually over-methylation of the nucleus can be triggered by piezo 1 type stress sensors, stress sensing ion channels in the plasma membrane. And so we have a chemical method to observe those to see the cascade of calcium signaling, calcium signaling to the nucleus, and then over-methylation of the interior of the over-methylation of chromatin. So for metaphase chromosomes, we were always wondering what would happen when we changed epigenetic marks on chromatin, returning to metaphase chromosomes, the very last thing I'll mention. And Ron Biggs did an interesting experiment whereby we expected hyperacetylation to make very soft metaphase chromosomes. I won't show the data, but very little happened with hyperacetylation. But hypermethylation, shown here, both monitoring of H3K9 and H3K27 methylation, leads to a big change in chromosome mechanical stiffness. The doubling, the force needed to double the length of the chromosome jumps up. That's not with like maybe some change in maybe the cell cycle is slow and there's a lot of SMC2 on the chromosome. No, actually the amount of SMC2 is about the same or even a little bit less. So in very recent studies now on bioRxiv, that's the same paper I was talking about a minute ago. Okay, so we can see that metaphase chromosomes are reduced in stiffness by removal of HPON alpha without methylstat treatment. So, and we can rescue that to some degree by treating with methylstat. So it does appear that HPON alpha, which we can see on the metaphase chromosome, is also playing some role in compacting and stiffening metaphase chromosomes. Interestingly, the HPON alpha degraded cells show a higher incidence of metaphase misalignment and missegregation of metaphase chromosomes, suggesting that really that H1 alpha is important in making sure that mammalian chromosomes are robust enough, well packaged enough, compacted enough, so that they can survive, properly perform and survive. Okay, so what I've told you about today is that chromosomes are geometrically and topologically organized, and thinking about both of them is some kind of a thing for biophysics people to work on. And for metaphase chromosomes, loop extrusion by SMCs without any condensation at all is sort of enough to drive this elongated chromatid and separated chromatid phenomenon that we see during cell division. And we're not at all thinking about chromatin as being highly condensed during metaphase. And I told you a little bit about metaphase. Thank you very much.
Thank you, John. We start with one question. So regarding also related to your last work on HP1 on the metaphase chromosome. So would you say that the role of HP1 is to add a bit of condensation in the compaction of your metaphase, or it's something different?
Well, so yeah, so so what we can have in polymer systems is we can have attractive interactions which are too weak to cause the sticking together of free polymers, but strong enough to affect already compacted or confined polymers. And so under the condition of high crowding of the chromatin in the metaphase chromatids, it does seem that HPO alpha, it's there and it's doing something to presumably provide physical crosslinks between chromatin fibers. So there's a lot of interesting questions to think about regarding that.
And when you do have loss of HP1, do you see a change in acetylation, for example, of chromatin that could also explain the differences in local chromatin
That's
organization?
a good question. I don't remember if we looked at that because we're very concerned with acetylation at that point. So I'd have to look back at the paper myself to answer that question. Thank you. But
Okay.
papers on bioRxiv, so we can have a look at it.
And there is
I don't think we did a panel like that, but I'm not 100% sure.
Okay, so we have a question also from Cedric, who is asking if you have any idea about the effect of Mg2 plus, because I think it's known that concentration of Mg2 plus is rising just before mitotic compaction. So, and we know that Mg2 plus may have some effect on the DNA.
Oh yeah, that was one of the first experiments that Michael Poyer did, was to study ion effects on compaction of chromosomes. And indeed, divalent metals, magnesium and calcium, just by themselves, are able to cause appreciable compaction of the chromosome. And it's kind of interesting. Divalents show a kind of bimodal behavior at low concentration. They will, presumably by bridging negative charges, compact chromosomes. But if you go to high concentrations of divalents, they'll start to screen, and you see actually relaxation and unfolding of chromosomes. But at concentrations we would expect in the cell, they can cause mild compaction, and I think it's well established in many experiments stretching back many years that's going on.
Okay, so we have another question from Geneviève Forel. Is HP1 alpha that you see on the metaphase chromosomes are on the arms or at the kinetochores?
It's concentrated at the kinetochore, but we do see it along the arms of the chromosomes as well. You do see it along. There's a kind of a dim staining along the arms of the chromosomes. So we are also doing experiments on the centromere these days, and because we know the arms are much less stiff than the centromere regions. So when you do this type of experiment, you're actually not very sensitive to centromere stiffness. The stiffness you get out of the whole chromosome is a weak spring. Imagine tying two rubber bands together, one very stiff and one very soft. When you stretch the two together, you stretch the soft one. You feel the spring constant of the soft.
Okay, so we have another question from Laura Cacciani. Thank you for the talk. How do you think mitotic chromosome linear compaction cope with sister chromatid cohesions?
Can you say it again? I did it. The sound went bad there for a second.
How do you think mitotic chromosome compaction cope with sister chromatid cohesions?
So, as you guys know, by the time you get to metaphase, so is the question more about metaphase or about prophase?
Yeah, that's a good question.
That's so at metaphase, along the arms, most of the cohesin has already been lost from the chromosome. We see that very clearly in experiments with, again, GFP cohesin fusions. But during the earlier parts of prophase, actually having cohesion between the sisters is a good idea because they can push each other apart by the internal chromatin loop pressure. But at the same time, it's a good idea to make those links go away as you go towards metaphase. And it appears that the natural selection has figured out those good ideas as well. So I don't know. Maybe the person wants to ask a follow-up question.
We have another question from Ranjit Pidantiri. So
oh,
John,
yeah.
Very nice talk.
I know Ranjit. Yeah, sure.
When you measure nucleus stiffness, do you fix the nucleus?
Oh, no, Ranjit, never. We never fix anything in our lab. Well, sometimes we do, but only for immunofluorescence. No. Yeah, so these preps of nuclei are native preps, where there's no fixation that's being done. Now, you could say as soon as you take stuff out of the cell, there might be some chemical effects that would effectively fixate some things, and that's certainly possible. But the isolations we do are fast in a matter of, minutes or even tens of seconds before the experiments start, before the mechanical experiments start to be done. And there are no fixatives used. The nucleus is removed from the cell interior to the cell exterior. What I pointed out briefly is that Drew did very nice experiments. He was actually able to measure the mechanical stiffness of nuclei inside intact cells. And so you can actually grab the nucleus in an intact cell by grabbing a plasma membrane, kind of, you can imagine having something that's surrounded by an envelope or a bed sheet or something. You can grab a beach ball through that membrane. And he was able to do experiments like that and saw similar spring constants as for isolated nuclei.
So we have a question from Olivier Cuvier related to your first parts.
Yes.
So is the condensing in the condensing dependent stretching, does a block in topo 2 decatenation impairs the stretching, the condensing related stretching?
Yeah, that's an excellent question, and we have not done that experiment. So that's an excellent experiment. I'll just show the.
And
yeah, no, that's an excellent suggestion. No, we have not mixed topo 2. It's a very logical thing to do. We have not mixed topo 2 and condense.
Okay. So another question from Olivier: can you measure stiffness in senescent cells where chromosomes detach from nuclear periphery?
Oh, that's another great question. And also, in senescent cells, you tend to have a lot of irregularities in the nuclear shape. And so that would be extremely interesting to look at. And it certainly can be done. That's certainly a doable experiment. And we have not done that. I was just going to pull up the.
a question from Nikolaos Araptisas. What physiological and functional outcome could have the alteration of the physical properties of the nucleus?
Yeah, good. So what we see is that this modulation of heterochromatin is playing a role not just in control of gene expression, which is classically how we think about heterochromatin, or at least maybe modern classically, but impressionistically. But also, heterochromatin is playing a key structural role in protecting the nucleus. And so we see very clearly that impairing heterochromatinization leads to destabilization of the nucleus. And I didn't show data, but Drew did nice studies showing, as other studies have shown with other types of nuclear envelope defects, for example, involving lamins, that when you start to get holes in the nucleus, you get a tremendous amount of DNA damage because you have cytoplasmic nucleases, which are able, it's always a bad idea to mix the nucleus contents with the cytoplasm contents, but in particular, there are nucleases which will start to cut up the DNA, and you very quickly see sites of DNA damage repair appear after a bleb occurs or a rupture occurs in the nucleus. So this alteration of heterochromatin levels can have a profound effect on DNA damage and then, other types of genetic mutation of cells. So this type of destabilization of the nucleus can lead to DNA damage and genome mutation. So it's very important to self-protect their contents. And the people that put cells through microfluidic constrictions have recently, like Jan Lammerding, have recently reported that they do see upticks in heterochromatinization associated with squashing of the cell and the nucleus. So it does appear that the cell has natural regulatory pathways that respond to it being squashed and the nucleus being squashed to try to protect the nucleus by heterochromatinizing parts, additional parts. So this, what we imagine is that heterochromatin, and heterochromatin becomes more and more important as you go to larger and larger genomes, which with larger and larger nuclei, which are more and more prone to being damaged as the cells crawl around and go through their daily lives. And so the picture is that heterochromatin is playing the role of a kind of a protective material for the genome, and that you need a lot of it to protect the genome when you have a large genome. And that it is also modulated in response to external stress, external mechanical stress on the cell.
Another question from Nikolaos. Do you observe heterogeneities, for example, in your stretching experiments?
Very much so, yes.
And what do you have any idea about the consequences, maybe physiological consequences on the nucleus?
Yeah, like everything else in the cell. Yeah, I'll just show you, remind you of Drew's experiments where I think this was very clear. When he showed, I have a couple of slides where all the data points are shown. This is the heterogeneity, say, in well-type nuclear stiffness. Now, part of that is variations in how we grab the nucleus and, how exactly experiments end up in a particular case, and probably the precise location of the cell in the cell cycle. We're synchronizing the cells by looking at nuclear size, basically identifying what we think are G1 nuclei, but they're probably at different stages of G1. And so you can see that this heterogeneity is quite large, over a factor of maybe 2 or so in stiffness. And so we have to do a number of experiments to get reliable statistics. And so that variability is undoubtedly connected with, as I was saying, to position in the cell cycle. There's more variability there in the HB1 alpha experiment. But also, yeah, probably just cell-to-cell variation in how exactly the nucleus has been organized. And yeah, it's very good.
And can you relate basically this? I mean, can you basically measure at the same time the mechanics properties and the HP1, whatever repartitions or cell cycle exactly where you are and correlate this type of information?
Yeah, in principle, those kinds of experiments can be done. And I mean, here's imaging of a rather weak HP1 alpha GFP signal in these cells. And so, yeah, we can try to get an idea of what's going on with redistribution. for example, HQ1 alpha or other proteins. Yeah, sure. Yeah, the experiments we do are, it's basically a question of how our collaborators genius in making genetically modified cell lines with could be multiple colors. We typically have a reporter for the gene we're degrading, for the protein we're degrading. In this case, H1 alpha I think is coupled to a red fluorescent. I think it's green fluorescent protein in this case. And then in addition, we typically have some other color protein providing a kind of secondary marker. So, two color experiments where you can look at, distributions. And we're using relatively poor wide field microscopy. This could all be done with higher tech microscopy. It's a question of technology.
Okay, so we have two last questions. So one from Geneviève. So she's asking, so phosphorylation is claimed to inhibit internucleosome interactions. However, H3 phosphorylation of serine 10 has been claimed to play a role in chromosome compaction or condensation. I don't know.
Yes.
What do you think?
What do I think? Well, as you know, H3S10 phosphorylation is clearly occurs clearly under cell cycle dependent kinase control. And I would say what it does remains one of the biggest mysteries in eukaryote cell biology. And yeah, I mean, I guess I've heard, I'm not a big expert on it. We haven't studied it in a lot of detail, but I will tell you one or two things. So H3S10 phosphorylation, I think some people have done experiments that suggest that it may have some interaction with condensate in some way. I forget the details. So I think there are experiments out there that suggest that. And probably there are people in the audience that are far bigger experts on this. But I will tell you one thing I do know. So we were very excited when we, at some point, took a metaphase chromosomes. I think in that case they were newt chromosomes. They might have been human chromosomes. And they're nice rubber bands, and we can hold on. And then we got out an enzyme called lambda phosphatase, and lambda phosphatase will dephosphorylate anything. Okay, so it's just like the cookie monster of phosphatases. It eats all phosphates. And when you spray that stuff on a metaphase chromosome, it just completely explodes. Okay, so that's taking away all phosphates on all proteins. It just disintegrates, right? It was really remarkable. So we said, okay, well, that's lambda phosphatase. Maybe it would be good to try to do something a little more specific. And so we started looking around for cell cycle controlled phosphatases that could be used in an in vitro setting, an in vitro experiment. And at the time we were doing this several years ago, really there were two candidates. One was something called peanuts. Yeah, so basically other, I think PP2A phosphatases, and yeah, PP2A turns out to not do anything unless it has a recognition protein associated with it. So these things always were complexes, as I recall. And so there was a complex called peanuts, which was supposed to be active at the end of mitosis to disassemble metaphase chromosomes. And so we got a small amount of that stuff, supposedly for in vitro study, and it didn't do anything at all. And then we also wanted to do experiments on the Earnshaw lab, and Paolo Bagnarelli have done a lot of work on something called RepoMan, that people are probably familiar with in the audience, which is another phosphatase complex. And there's been, I think, a long effort to try to do something in vitro to get the stuff as an in vitro reagent, but that's not available yet. So we have good evidence that dephosphorylation can profoundly modify metaphase chromosome structure, but we don't have a very clear idea of how.
Okay.
Or any idea of the specificity. What could be, maybe we hit H3S10 and the whole chromosome blows apart, or it could be we're dephosphorylating.
Is there any, specific dephosphatase for serine 10 or?
I don't think it's known. I mean, people will correct me in the audience if I'm wrong, but I don't think that's known. I don't think it's known.
Okay, Genevieve asked, did you try PP1, which is supposed to end mitosis?
Yeah, I forget all the details of those experiments there several years ago, but yeah, I think we did, and it was not helpful, not what do you say, informative. PP2A and PP1, I think were the guys.
Okay, and maybe a last question from Cedric, a general question. So, what's your feeling about what controls the size of the nucleus? I mean,
Well, that's a good question. You know, the talk we heard earlier about C. elegans, you know, I had a recent theory student, Sumitava Brahmachari, who was doing this polymer brush modeling of chromosomes. And along the way, he sort of made a library. You can find it, the data in, I think, the supplementary information for the paper that was published by Sumitava on this. And, you know, C. elegans has a particularly kind of low concentration of chromatin in its nucleus, is what we concluded from data that's available in the literature. And also, budding yeast has a pretty low chromatin fraction compared to, say, a mouse or a human nucleus, which is still fairly low. It's in the, I think, 10% range, volume fraction. The elegans and budding yeast are rather low. So it does seem that, you know, the size of the nucleus is a kind of adjustable parameter. And one thing that's interesting about budding yeast is it doesn't really have the same kind of chromosome compaction machinery at work or necessary for separation of most of the chromosomes, mainly the nucleolus. The arms seem to be separated without the action of condensin. And that may just be that that's a strategy. Make a big nucleus, and then it's easy to separate your chromosomes from one another. I was fascinated by the talk with the knockdowns of cohesin because it suggests that, yeah, the cells can sort of get along without cohesin. And maybe it's just because the chromosomes have a lot of room to move around and are able to disentangle reasonably well without the action of those complexes. So I think the size of the nucleus is a kind of free parameter that you can use to control what other strategies you're using to compact chromosomes from a, you know, from a kind of a evolutionary perspective.
but even in human, you have a high variability in terms of
Yes.
your
Yeah.
size. I mean, if you go from
Yeah.
neurons to an embryonic stem cells, I mean,
Yeah. Oh, yeah. Yeah. Right. Through development. Yeah. Sure. Yeah. Yeah. Sure. Absolutely. Right. So, I mean, I think this kind of just reflects different strategies that can be used at different times for making sure the chromosomes can come apart during mitosis.